This is podcast number 39. In this podcast, we're going to circle back and look at how you digest other things besides carbohydrates. So we spent a lot of time talking about how you went from the lactose in milk to glucose and galactose, and then glucose all the way down to get ATP out of glucose in stage 1, 2, 3, and 4. But there's certainly other things than glucose in milk. What about protein? What about lipids? Here's generic amino acid. They have in the milk protein casein. There are 225 different kinds of, or 225 amino acids, not 225 different kinds, 20 different kinds, but 225 amino acids. This is the one-letter code for the primary sequence of casein, methionine, lysine, valine, leucine, isoleucine, leucine, etc. So how would you digest this sort of thing? 224 different peptide bonds, over 450 carbons, including the R groups. And there's also lipids in fatty acid. Palmitic acid is a common lipid that we have in our food. It's a straight chain fatty acid. Another one that is common is oleic acid that has one double bond along the chain that gives the chain a kink. Oleic acid is common in olive oil, for example. Palmitic acid is common in animal fats like butter. So this would be a fatty acid you would find in milk. And how do you break these things down to get ATP? We talked about in lactose taking the glucose and the galactose apart and then taking glucose all the way down to its components to generate ATP. And we'll talk about, in general, fatty acids and, in general, proteins. Okay, so if we talk about lipids as a type of fatty acid, how do you oxidize this? How do you get energy out of this? Convert the energy in all those bonds to ATP, which is more easily used. So just by looking at this molecule, you can tell that there's a lot of bonds in there. And in fact, it's the most efficient way of storing bond energy is in a fatty acid. And this is part of the reason why we store excess energy as fat, is because in a very small amount of space, you can get a lot of potential energy stored in those bonds. That means that we aren't so big. If we stored everything as glucose, for example, in glycogen or something like that, that would be 8 to 9 times bigger amount of volume. So most of us don't want to be 8 to 9 times bigger than we are. So fat gets a bad name, but it's a very efficient way of storing excess energy, that it doesn't take up a ton of space in there. Okay, so if we use palmitic acid as a type of fatty acid that you would burn, it has 16 carbons in its fatty acid chain. 1, 2, 3, 4, 5, 6, 16. Okay, this is 8 groups of 2. And those groups of 2 can be added 2 at a time to the coenzyme A molecule, and then it can be taken through the citric acid cycle. So you'll remember that at the end of glycolysis, you ended up with a pyruvic acid molecule in stage 1, and then in stage 2, you remove one of those carbons, breathe it out as CO2, and you take those 2 carbons and attach it to CoA. For a fatty acid molecule, you can take off 2 carbons at a time and attach it to CoA. And you do that 2 times for glucose, but something like a fatty acid like palmitic acid, you do it 8 times because there's 16 carbons. You also have fatty acids that are 22 carbons big. So you can start to get the idea that fatty acids have a lot of energy in them. And then each time you take off 2, you go through the citric acid cycle. So in the transfer of those 2 carbons to the acetyl group, you don't transfer oxygen. But when the fatty acid is completely oxidized, that 2-carbon molecule goes through the citric acid cycle, and then the NADH and FADH2 that you generate goes through stage 4, the electron transport chain, and then stage 5, you convert it to ATP. And you do that for every 2-carbon molecule you pulled off the fatty acid. So one fatty acid molecule, you're going to go, if it's a 16-carbon molecule, you're going to go through the citric acid cycle 8 times. And then you're going to make lots of FAD reduced and NAD reduced, and then you're going to pour all that into the electron transport chain, move lots of protons into the intermembrane space. Lots of protons will fall back from the intermembrane space into the matrix, making lots of ATP. So after you make that 2-carbon molecule, it's going to get added to this 4-carbon molecule and then go around the citric acid cycle. That's stage 3, stage 4, stage 5, just like you saw before with glucose. So in the 80s, the idea of aerobics became a thing. This is before most of you were born. But the idea that you had to have low impact, high repetition exercise in order to burn fat. And it was called aerobic because aerobic means with oxygen. So the type of exercise you're doing, if you can keep it low impact, high repetition, then you can keep your muscles supplied with oxygen and allow your muscles to go through stage 3, 4, and 5, and then be able to break down fat. If you can't go through stage 3, 4, and 5 because you're using oxygen too fast for your cells to go through stage 3, 4, and 5. Oxygen, remember, is required at the end of stage 4 to accept the electrons that are coming down the electron transport chain. Then you can't do stage 3 and 4 and 5, and then you can't burn fat. So people that are interested in losing weight stored in fat need to do low impact, high repetition exercise. Running, for example, or aerobic, like aerobics in an aerobics studio. These are all relatively low impact, high repetition exercises that keep oxygen supplied to the muscle and allows the muscle cell to go through stage 3, 4, and 5. If you are using energy faster than you can supply oxygen to the muscle, this would be something like sprinting or lifting very high weights, very heavy weights. Then the muscle is using energy faster than oxygen supply can keep up with. That will not use oxygen during that procedure while the muscle is going through that procedure, and therefore they are not as recommended for someone who's trying to lose weight through exercise. Now, after you recover from the high impact, low oxygen sort of exercise, you're going to use oxygen to reoxidize the lactic acid you make up. That's a different story. But in general, the reason why aerobics is called aerobics is because you exercise while supplying oxygen. And if you want to burn fat, you need oxygen. And this is why, is that you need stage 3, 4, and 5 Stage 4 depends on oxygen. Stage 4 will back up into stage 3. And stage 1 in glycolysis won't burn fat. You need stage 3, 4, and 5 to burn fat. Okay, how about proteins? Okay. So proteins are broken down by enzymes. Enzymes are going to attack the peptide bond between amino acids in proteins, and enzymes have different characteristics. For example, enzymes typically work at a given pH and not at all pHs. So if we look at pH scale on the x-axis here, this would be a very acidic, this would be very basic. Remember, it's a 10-fold difference in hydrogen ion concentration for every 1 fold of the pH scale. And then we just look at how much activity you see for a given enzyme. For enzymes that live in the stomach and work on stomach proteins that arrive in the stomach in our food, they have a activity enzyme that is at a very low pH because the pH of your stomach is really low. That's thought to have evolved to aid digestion. You can mechanically break some of those bonds with low pH, but also it's antibacterial in that a lot of the bacteria that we would ingest can't survive that low pH. And so an enzyme that can work in that environment, which is not a typical cell pH, is advantageous. If we look at enzymes that work inside a cell environment, things like trypsin and chymotrypsin, these are proteases, enzymes that cut proteins that work inside a cell environment. They work at more typical cell pHs, okay? And this one, these are found in the intestines, which is more typical of a cell pH, although pH 9 is getting pretty high. There's something that happens in digestion where the pH of the the pH of the food environment where food is digested can change. Maybe you get into that in Principles 2 or in another physiology course. This pH dependence on how enzymes function actually came up recently in drugs that were trying to fight the COVID-19 epidemic. So the hydroxychloroquine, the drug that was advanced by the White House as a possible cure or drug that would help in COVID-19 infections, it works by inhibiting proteases. So when the virus enters the cell, it makes a very long viral protein that then is cut into smaller bits, and those smaller bits are important to reassemble new viruses. So if you can stop it from being cut into smaller bits, then you stop replication of the virus because that enzyme is required for replication of the virus. The drug hydroxychloroquine changed the pH in a compartment inside the cell, making it less optimal for that enzyme to work. However, that one hasn't panned out, and that one is now off of clinical trials and is no longer prescribed, are no longer recommended anyway for people suffering from COVID-19. Okay, so can you take one enzyme that digests proteins and will digest all proteins? So pepsin that digests at an optimal pH of 2, give it 3 different proteins to digest, see how it does. So if you take a protein from egg whites, ovalbumin, it doesn't do great, it does okay. If you give it another protein to digest, like hemoglobin, it does much better. And if you take hemoglobin apart without the heme, it does a little bit worse. But the point is that not all proteins are equally well suited for that particular protease to work on. and that's part of the reason why you have multiple proteases. You know, it's not one protease breaks down all proteins. Okay, so let's say you found a protease that can digest your protein and cut it up. One of the things it can generate is a 2-carbon intermediate. Here's the enzyme GDH. Can produce this. This is another kind of protease. It can produce this 2-carbon intermediate, and hopefully you recognize this by now. This 2-carbon intermediate is key to adding that to coenzyme A and entering stage 3. So you saw that in breaking down carbohydrate in glucose. You saw that in breaking down lipids in palmitic acid. And now you're seeing it in breaking down proteins, in this case from an amino acid. And then once you enter stage 3, you can go through stage 4 and stage 5. So you generate a 2-carbon intermediate that's added to oxaloacetic acid in the Krebs cycle, makes 6-carbon acetic acid, and then you go through the stage 3, 4, and 5 and get ATP from breakdown of protein. Again, breaking down protein would require oxygen because stage 3, 4, and 5, 4 in particular, requires oxygen. And if 4 doesn't happen, it backs up into 3, and then 3 doesn't happen. Okay, so if you're going to take 2-carbon units off of proteins, how do you regulate that? So there's that enzyme GDH that we saw in the previous slide is a protease. How do you regulate that? Well, is it regulated by ATP? It does not look like it's regulated by ATP in that you don't get much of a change in how much NADH you produce from this reaction. Let's go back to this. So this reaction, GDH produces the 2-carbon intermediate, which goes into stage 3, and also produces some NADH, just like we saw in stage 2 of carbohydrate breakdown. So if we add ATP, we don't get much NADH produced, and if we it doesn't seem to make, the line is pretty flat. But if we add ADP, okay, you get a pretty steep curve, and you get NADH increased. So when ATP is depleted, less ATP, and you need energy, that's when you can start breaking down protein to make ATP. What do cells choose if they've got a choice of lipid to break down, or protein to break down, or carbohydrate to break down? In general, carbohydrate is the first choice, but that's also depends on the organism. So organisms like fish, like salmon, that go on a very long migration, they will stop eating during that migration, and they might not eat for 11 months. 11 months in a row, no eating. And they're exercising the whole time that they're not eating. They're swimming hundreds of miles in the ocean, getting back to the breeding grounds while they're not eating. And you've seen all those movies of salmon swimming upstream. Those animals are preferentially breaking down protein. They are breaking down lipid and carbohydrate, but they are breaking down protein preferentially. In humans, if you starve a human, they will go through their carbohydrate reserves typically, but then they don't burn through all their fat, because fat is not just energy storage, but fat, you also need fat to make hormones, signaling molecules, that sort of thing. And so even if a person starves to death, they will still have some fat on them. It doesn't completely go away. And so, and of course, protein, your brain is has a lot of protein in it. And if you start digesting your protein, you can have neural consequences. In fact, you have neural consequences from any kind of starvation. So it's not that you have 3 depots of energy and you completely get rid of one before you go down to the next. You're going to use all of them all at the same time. Different organisms have different priorities in terms of what they're going to tap first versus second. In general, carbohydrates are tapped first, but it depends on the organism. ATP is not the only repressor. If you look at the effects of ATP on this protease, it is less severe suppressor of that protease than is GTP. And as we mentioned when we talked about citric acid cycle, GTP is produced in the citric acid cycle by animal cells, and it's a pretty potent inhibitor of deamination. So if there's lots of GTP around, it's unlikely that the cell is going to break down more protein in order to make GTP, but it could be breaking down other things in order to make GTP. But if there's not much GTP around, that's likely because carbohydrate reserves have been depleted and lipid reserves have been depleted, and so maybe now it's a time to dip into the protein reserves. Again, that depends on the organism.